Anyway, I am very, very pleased as president of SSE to introduce Sally Otto. Actually, Sally, and I don't think I know you personally, but I've seen you around at meetings. I think we've met a couple times. I do know that Sally has had a strong presence in SSE. She's been president of our society. She has done a number of, she has had a number of different accolades, including sort of a reputation as a person who is no nonsense and contributes to betterment of her surroundings. She is a member of the National Academy of Sciences and also just a very productive scientist. So I'm not going to go on much longer since I wasn't expected to do this, but I'm really looking forward to seeing your talk, Sally.
Very well for an impromptu introduction. Thank you, Kelly. And you know, I have to say it is such a pleasure to receive this award because it's from my peers, and even on this call, seeing familiar faces from all sorts of different stages of my career. It's just a real honor to be here today. So let me just get my Zoom settings the way I like them. All right, and so what I wanted to do today was to go over some of the work that I've been involved in across the pandemic over the last 3 years as SARS-CoV-2 continues to evolve. And as we all know, when the virus was detected and this new respiratory illness was emerged in Wuhan, China in the end of 2019, right from the very beginning, the sequence came out, and right from the very beginning, scientists were searching for signatures of evolution and adaptation to humans. And since that time, we've gathered an enormous amount of data. Never before have we had so much information about evolution as it unfolded, about any population of viruses or what have you. Here's yesterday's GISAID count tally of 15 million genomes of this virus to date, which is giving us an incredible insight into evolutionary processes. And I want to reflect on those today. But before I do, I really want to shout out, because this community, the evolutionary community, so many individuals within this community really stepped up to the plate. And in the face of a pandemic virus that we knew really little about, we didn't know how it was transmitted, we didn't know how often it was transmitted, we didn't know what types of selective changes it might be undergoing. And this is just a small spattering of the evolutionary biologists that pivoted and spent a lot of their research time over the last 3 years helping our community build knowledge about this virus. So thank you to them. And this represents the many of you that I haven't grabbed your picture, because this is only the tip of the iceberg. So thank you. And I think our community did a really fantastic job of stepping up. I was involved in 2 groups with a focus in Canada. One was in the British Columbia, the BC COVID-19 modeling group. And again, a really high caliber set of folks got together to contribute different kinds of modeling to the effort to understand what was going on locally and to provide policy advice, to model in advance of a policy decision. So thinking about what was coming down the line, down the road soon, what decisions had to be made, and what information would be needed for those decisions. And we're continuing to do this at a Canada-wide level as part of the COVARNet network. This network is very interesting. It's a network of scientists that includes EDI components, social scientists, molecular and cell biologists, virologists, public health experts, and evolutionary biologists. And so I think learning from each other has been a really interesting way to go and do science jointly. So how we do science, and my experience of how I'm doing science, has really changed through the pandemic. And it's partly these collaborations, but it's also the fact that when things are changing so fast, we can't wait for the regular publication cycle. So bioRxiv has played such a big role, as has Twitter. But then those aren't peer-reviewed yet. So there's kind of this fast peer review process that happens often through Twitter, but then also through technical briefings and these groups of scientists working together and playing that peer review role on the fly. So let's get into it and dive in. What did we learn about this virus and how it has evolved? This is a picture of its evolution from the first year. This is from NextStrain. Every dot is a collection of a different individual sample, and these concentric rings are the dates those samples were taken from. And you can see a few things. The common ancestor coalesces in late 2019, exactly as we expected from that that introduction in Wuhan. So there wasn't previous circulation deep in evolutionary time. This was everything coalesced to about that time. We also can estimate the rate of mutational changes by how many differences occur per unit time through as we move out on this circle. And that's about one change in the genome every 2 weeks. But the key thing I wanted to highlight in this first graph is just how star-like this phylogeny is. And the growth of SARS-CoV-2 population across the world happened in a way where every lineage was almost equally likely to lead to descendants. And that's what leads to this star-shaped phylogeny, a growing population with pretty much similar chances of descent. I'm just going to check the chat to see. Okay, no, that's good. So the first year then didn't really show many signs of selective sweeps. And that is in stark contrast to what we saw in the second year. So the second year of the pandemic started with this picture from Theo Sanderson pulled this graphic together of the spread of alpha B117 in the UK. And in health district after health district, and in week after week, the frequency of the alpha variant was rising. And this was the first super strong evidence of selection because it was repeated in time and location. Often it's hard to tell, did this variant go up in frequency because there was an outbreak or a big migration event? But this was clear evidence of selection. And indeed, you see the signal of that in the phylogeny sampled after that time. It's a little hard to see because it's been squished now, dwarfed by, but there was an alpha wave that spread and dominated sequences. Here you see the delta wave now dominating the sequences in 2022, and then the Omicron sequences coming. And so not every lineage has equal chances of success in spreading. We're seeing some variants outcompeting others. And the third year has been more of the same with BA1 and BA2, BA5, BQ spreading here and taking over, and most recently XBB spreading and taking over, dominating the evolutionary tree of this virus. So I'm going to talk about some modeling in the kind of three phases of this pandemic. The early phase where there was a largely susceptible population, and then the rollout of vaccines. And finally, I want to reflect on evolutionary pressures on the virus in the present when there's waning immunity. So modeling. The type of model that is constructed in epidemiological evolutionary epidemiology are these SIR type models. So S for susceptible, I for infected, R for recovered or resistant. And these compartment models track transmission events from susceptible to one of the infected classes, as well as recovery. But this particular model that we did in 2020 was tailored a little bit for SARS-CoV-2. It allowed for the possibility of asymptomatic transmission. There was uncertainty at the time of how much asymptomatic transmission there was, but that class was known to be about a third of infections led to asymptomatic transmission, as well as substantial pre-symptomatic transmission before becoming symptomatic. So we can we can add mutations to this model and see what happens to the fate of a variant. Early on, there were lots of infections, but the vast majority of the population remained uninfected. The number of susceptible individuals was roughly constant for over the first year, roughly at the same high number. And that helps you do the math, because if you can treat that susceptible pool as kind of constant, close enough, then all of these equations turn into a linear set of equations. And then you can track how the numbers of a virus change over time. And it turns into something a lot like a demographic model of an age-structured population. But in this case, instead of ages, we have stages, the fraction of the population of viruses that are in each of these stages, as shown in this graph here. And we can then see if a mutation arises that changes the life history of this virus, makes it more transmissible, or maybe makes it stay in the exposed category for longer or shorter. What happens to that variant? And we can use math to estimate what the selection ought to be on a new variant, depending on how it changes the growth rate as measured by lambda, the leading eigenvalue of this series of equations as captured by this kind of demographic matrix M. And when you crunch the numbers, you get this kind of complicated formula, but I want to walk you through and get the insights that we can glean as evolutionary biologists from this selection coefficient. So first of all, the mutation has to change something. And so these delta terms are the various things that a mutation might change. It can change any of these life history parameters. It can change the betas, which are the transmission rates, or could maybe change the mortality rate or the clearance rates, these kappa values. So those are the mutant effects. There's other terms in this equation that are used. Those U's are the proportion of the virus in each of these stages. Much like a stable age distribution, this is the stable stage distribution for how much of the virus at any point in time is in asymptomatic infections, exposed class, etc. There's one last thing I have to describe, and that is these V's. These V's are describe the reproductive value of an infection in each class. The value in demography, and here too, is saying what fraction of the population in the long-term future descends from an individual in this stage at the moment. So VP would be what's the fraction of the population expected to descend from a virus in the pre-symptomatic class versus VI in the symptomatic class. And we know something about those reproductive values. We can show, for example, that the pre-symptomatic class always has a higher reproductive value because you get all of the infections from that stage, and then eventually they move to the symptomatic class and also have infections in that stage. So this is always a positive quantity. As a consequence, we can really dive in and figure out how a virus like SARS-CoV-2 is expected to evolve. Some things are pretty obvious. For example, transmission rates it's always selectively favored. All of these things are positive. It's always selectively favored for the virus to evolve to be more transmittable. And that can either be through intrinsic transmissibility, the virus gets into our cells more quickly, or has a higher viral load and so is more likely to infect another person, or it could be through higher immune evasion and so transmit to more people. So that's a possibility too. The other feature of this evolution is selection to decrease the exit rates. And that makes sense too. The longer that the virus stays infectious in a person, the more selectively favored that would be. And so the other component of selection on SARS-CoV-2 is for prolonged infectivity. And there also can be shifts to a longer exposed phase or a shorter exposed phase, but that kind of depends on whether or not cases are rising or falling, kind of more of a detail. But one of the biggest take-home messages, I think, from this model was this part, that there was negligible direct selection on severity and mortality. That is, when we crunched the numbers, there was almost no selection on this virus to become more or less deadly. And the reason for that is shown in this kind of life cycle diagram of what happens to the viral load within your body. And the viral load skyrockets and declines over the course of days, pretty much long before that critical phase of severe disease, hospitalization, and death. And so as far as the evolution of this virus is concerned, its evolutionary fate is decoupled from what happens to the patient it infected. It's not evolving to kill you or not to kill you because it has left your body before those severe sequelae happen. And that's why for SARS-CoV-2, there's almost no direct selection on severity and mortality. And this contrasts, other diseases aren't like this. Other diseases kill you during this acute phase of a high viral load. And so whatever the virus can do by evolving to reduce severity is an interest, but that's just not true for SARS-CoV-2. And indeed, in this paper, so this is before the first variant arose, we wrote that there wasn't direct selection, but there were many plausible routes of indirect selection that would affect the virulence of this disease. So mutations could arise that would improve transmission rates by increasing viral replication, and that would lead potentially to a higher viral impact, more damage to tissues, a higher immune response, and potentially more deaths and severe cases. So that's a case of positive pleiotropy. The virus gets better at replicating, and as a consequence of that replication, it transmits better, which is what is selected. But in addition, it tends to hurt us more. But alternatively, we noted it's also possible that the virus evolves to transmit better by changing the tissues in which it tends to dominate. The original SARS-CoV-2 virus would lodge deep in the lungs, causing a lower respiratory tract infection, which caused a lot of damage to the lungs and a lot of severe cases. But we argued that it could have evolved to switch which cells it preferentially infected. And if it infected individuals in the upper airways, that could have increased transmission as well and been selected for that transmissibility. But that type of mutation would have a negative pleiotropic effect and reduce severity by being more of an upper airways infection. Both of those were possible. The model didn't say which kind of pleiotropy could arise, but it said that it scoped out the realm of possibilities to say that both less and more severe mutations could spread due to pleiotropy. And indeed, what has come to pass are examples of many variants of concern that are more virulent and more deadly, including alpha, beta, delta, and to some extent gamma, due in part because of this higher replication rate, but then also variants like Omicron that are less deadly. And Omicron did this switch in tissue tropism to become an upper airways infection that is less deadly. So the models helped see what might be possible with this virus, and also cleared up this kind of common misconception that viruses will always evolve to become milder. So that's the selection coefficient that the model predicts. What were we seeing on the ground? What were we seeing in the genomes that were being sequenced over time? So I want to walk through this, how we measure selection, and to give you a sense of the process. So here's the type of data we have. You go into GISAID, and from a country, you can get the number of sequences on any given day. The size of the dot determines the number of sequences, and they are typed by their Pango lineages like BA2 and BA5. And so from that, you get a graph of the frequency changes for BA5 here rising over time and BA2 declining. So let's let P be the frequency of BA5. 1 minus P is the frequency of BA2. And selection, the evolutionary change that occurs or that's expected is proportional to the variance, PQ, P times 1 minus P, times the selection coefficient acting in favor here of BA5. So this is a standard haploid model of selection, and you can solve it to predict how the allele frequency should change over time. These are the same equations. This differential equation has this prediction for this time course of evolution. And what that allows us to do is that on any particular date, we know how many individuals we've sequenced. We've got a sample of size n, and j of them are BA5 and k of them are BA2. And this equation allows us to predict using this binomial formula what number of observed BA2 and BA5 we'd see on a particular day t, given this equation here. And the two unknowns, what we don't know, we don't have knowledge ahead of time of what the selection coefficient is, or what the starting frequency is, P naught. And so this has two unknowns in it. That's just for one day. And if we multiply the likelihood on one day times the next day, times the next day, times the next day, and actually log that, then we can turn this formula. We can drop all of these binomial coefficients because they don't even involve the unknown parameters. These are the only things that involve the unknown parameters. And you get an overall likelihood of seeing data like this. And from that, we can estimate selection over time and the confidence interval in that selection coefficient. There's another way that we can visualize this that provides even maybe some more insight. And so I want to go a step back. So that's great. That gives us a selection coefficient, but I'm going to move forward and rearrange this equation. It's called a logit, L-O-G-I-T rearrangement. And what you do is you take the frequency of the type of interest, here BA5, you divide it by everything else, here BA2. And when you take this and divide it by 1 minus p, you end up getting this equation. If you log it, then and expand out, that gets rid of the e, and you end up with a linear equation, s times t, a linear equation over time. As time passes, you expect a positively favored variant to rise in frequency on this logit scale as a line. And then the intercept of that line depends on the initial frequency, which makes sense. So this is that ratio of frequencies on a log scale. And what's nice about this is it gives you a way to even visually see, is a model of selection actually consistent with a rise in a particular variant? As I mentioned, sometimes it was hard to tell, was it just an outbreak or something like that? That can lead to a rise in frequency, but it doesn't lead to this kind of consistent rise over time with the same slope. Whereas if it's selection, we see something more like this consistent rise over time with a fairly constant slope. Now, interestingly, let's go back to the model. The model told us something that we should predict, and that is that the selective advantage is proportional to how many people are out and about, the susceptible class. But if we all stay home, there aren't susceptible individuals to infect. And so this whole selection coefficient favoring more transmissible variants should decline. And so what we do as a population should influence the selection that we see. And indeed, there are times over the course of the pandemic where we get kinks in this evolutionary graph. For example, as we saw in March of 2021, where the alpha wave was spreading so quickly that there was a circuit breaker in March and at the end of March here in British Columbia and in many other jurisdictions. Here we closed indoor dining, gyms were closed, travel within the province was restricted, and there was this just social pressure to isolate as best you could. And what we saw was this kink where the selection coefficient was stronger before the circuit breaker and then relaxed. Sometimes these kinks are associated with circuit breakers. Sometimes they're just associated with people becoming very concerned about what they're seeing around them. This collective concern was very apparent with Omicron, where we see these kind of rises in frequency on this logit plot with really strong selection, 25%, between 25 and 30%. So really fast spread of Omicron within the COVID population, SARS-CoV-2 population, and then this kink. But that kink corresponded not to a circuit breaker or anything like that, but media reports of lines, day-long lines to get tested for COVID, lines of ambulances outside of hospitals where people couldn't get into emergency rooms for hours or even for more than a day. And that concern about a collapse of the healthcare system led to much more careful behavior and this kink due to collective change in our behavior. But oftentimes, especially early on, that selection is fairly constant over time, and we can measure it. This is just a graph showing you what the selection coefficients have been over the course of the 3 years of the pandemic. And these are just the major variants. So alpha and gamma had a selective advantage of about 8% over the previous circulating wild type or Wuhan strain. Delta had a selective advantage over alpha and gamma of a little bit higher than that. And these black arrows are showing that when we measure selection, we're measuring it relative to the kind of previously common variant. So I've shown what we're comparing to what using these arrows. And so recently we had a sweep of BQ within a population dominated by BA5, and the advantage of BQ over BA5 was, again, approximately 7%. XBB, with similarly with XBB, which we'll see in a moment. So what does that mean, these selection coefficients? Let me translate that. First of all, to just kind of step back and say, these are selection coefficients per day. So as an evolutionary biologist, this is mind-blowing. When Omicron, when our first estimates appeared and we're like, it's over 20% selective advantage per day, that was unfathomable. It meant that this virus was doubling in relative frequency in a matter of 2 to 3 days. And I've never seen a selection coefficient like that in nature. But even these ones are doubling times on the order of a week, which is astronomically strong selection. So what about where are we going in the future? In order to keep track of which variants are spreading and how fast, we need a way to automate this. Because in Canada over the last 3 months, for example, there have been over 500 Omicron name lineages that have arisen. So there's a lot of diversity that's arising because there's a lot of cases. And we need to track which of those are worrisome and which ones we can ignore. So in the CoVarNet group, we've got a GitHub package called Duotang that does various things, including tracking variants. Here's what we're seeing recently. This is on a logit plot, but this is the regular frequency plot showing the rise to dominating SARS-CoV-2 of XBB.1.5. So that's the majority strain at the moment. We're seeing an uptick. There's actually a few different variants that we're tracking at the moment, but this 1.16 is the one we've got our eyes on the most. So what is this XBB.1.5 variant that is the majority strain now, both in Canada and in the United States? Well, it is itself a descendant of XBB. That X means it's a recombinant. So anytime you see a lineage that's called X, like XBC or XAY, those are recombinant strains. In this particular case, it's a recombinant of 2 BA2 parents. And so it's combining this mutations on the left from one parent and with those on the right from the other parent. And this combination of spike mutations has been shown to be highly immune evasive. So XBB as well as BQ strains have been shown in the lab to have really strong immune evasive properties. But XBB.1.5, so this is its background, but it's evolved new mutations. And in particular, one mutation, here's a one-step mutation, F486S. It's now changed in this XBB.1.5 to a proline from originally it was phenylalanine, and now it's proline. That's interesting to us because that type of amino acid change is a two-step mutation. It's not, there's no one single mutation in that codon that can take you from a phenylalanine to a proline. Only by having one mutation and then another in the same codon can you get this evolutionary change. So that's saying that now we're starting to see these not just kind of adjacent mutational neighbors evolving and spreading, but these kind of two-step neighbors. This virus is, there's so many variants and so much diversity is rising that we're starting to see these two-step explorations of the fitness landscape of the virus. And that particular mutational change has been shown to increase binding to the ACE2 receptors on our cells. So it has a higher transmissibility in addition to the background of strong immune evasion of XBB. So we scan all of these 500 variants for ones that are spreading really fast. Here are the ones that are currently spreading the fastest. The color denotes how many sequences we have. So the darker the purple, the more evidence we have for fast spread. And if it's spreading more significantly in more than one province, we give it a double circle. So we can really visually see which ones we have the strongest evidence of spread. And right now at the moment, that's an XBB.1.16 variant, which has another change that is thought to provide some immune evasive potential to it. We're tracking it as it rises in frequency. Since this is an evolution audience, I think it's important that we talk about what this measure of selection is and how different people and different groups are using different versions of this measure. So when I'm talking about selection coefficient, it's per day, and it's measuring the relative frequency change per day. Other groups have used per generation, depending on the generation time. Cove Spectrum switched recently to using per week. So if you go on to Cove Spectrum and look at XBB.1.16.1, which we were just looking at, it doesn't have a selection coefficient of 0.08. It has one of 42% or 0.42, but that's per week. And so you can't compare those directly. If this is per week, that corresponds to a selection coefficient of 6% per day or 0.06. So that's one thing to pay attention to. What is the time unit? And don't compare different groups without checking the time unit. The other thing to pay attention to is that what we do are very specific about what's the selective advantage relative to a specific strain that's co-circulating at that time. Here, for example, we're measuring the advantage of XBB.1.16.1 against XBB.1.5 that's co-circulating. So we know this is the selection of type A versus type B. And which one is outcompeting the other. Cove Spectrum and other automated sites often measure the selective spread relative to the rest, all of the rest. And that can cause some confusion because it can lead to selection coefficients that don't match a standard selective trajectories that don't match what you'd expect for selection, an S-shaped curve. And that's because the rest is changing. Here, for example, the BQ, it was rising as you'd expect due to selection and frequency, but then it started falling in frequency. And that's because the rest changed. XBB appeared. Now XBB is outcompeting BQ1 and driving this frequency down. And so on average, this is the model prediction, and clearly it's not obeying the model, but it's not obeying the model because the background is changing. And so just be aware that, and think about what is it that you're measuring the selection of. And then if you're measuring it against every other virus out there, and that virus is evolving as a group as well, then these selection coefficients become somewhat undefined. Okay, technical aside over. For policymakers, they're interested in evolution, but that doesn't matter so much as they want to know what this means for cases. And so we can combine that evolutionary picture for changes in allele frequency with what we know about trends in cases overall. The problem is right now, poor surveillance makes that challenging to do. So we actually don't have very good trend numbers for cases. we've been using the number of cases in individuals over 70 in age in Canada because that group is more consistently tested when they go to the doctor or hospital. So those are the green dots. And so that's the case numbers as they rise and fall over time. And we can multiply that by the frequencies, that PT equation I was showing you earlier, to get how the variants are changing and underlying those trends. And this is helpful. This is visually helpful because it allows us to see things like you can kind of see in this graph, where the cases may be declining and you see overall a decline in cases, but underlying that you can see the uptick or the rise in a particular variant. So that was happening back here, that BQ was declining, but we could see XBB rising in frequency, and it's happening again now, but one variant's declining and the next is coming up under it with a positive growth rate. So we don't predict the continued decline of cases because this next variant is on the rise. And that has allowed us as evolutionary biologists to tell policymakers, alpha is on the rise. And this is roughly when alpha will lead to an inflection in number of cases and cause cases to rise. Same with Omicron, same with Delta. So it's given policymakers a heads up about what changes to expect, what's the rate of growth that we're going to expect over the next few weeks. So that's the kind of phase 1 earlier models where we were focusing on a largely susceptible population. But of course, now we're in a different environment. The rollout of vaccines was an incredible feat in upper middle income countries. Here are charts of the number of people that are fully vaccinated, the 2 course series. So in Canada, it was astronomical rise over the summer of 2021 to over 80% fully vaccinated. And you can see that pretty much follows in upper middle income countries. But low income countries continue to have inadequate access to resources for vaccination and continue to have low vaccination rates under 30%. So as evolutionary biologists, some of how we can advise about vaccine rollouts had to do with what can we do to avoid immune evasion? How could we slow the spread of mutations that would evade the immunity provided by vaccines? Because it's not just about getting shots in arms. We want those shots to work as long as possible. And so we developed a modified SIR model that allowed in a mutant that was immune evasive to be able to infect individuals that were otherwise protected or recovered. So we allowed this mutant to be immune evasive to some extent, and infect individuals that were otherwise resistant. And crunching the numbers allowed us to say things like the time until we first expect an immune escape mutant should be given this formula, so we could estimate how long we would have to wait. And then we could estimate what the selective advantage of the new mutation is. And that then could be translated into policy advice. If you want to slow the appearance of the mutation, and I'm going to go through this quickly, you have kind of 2 things you can do. You either reduce the mutation rate. How do you do that? You avoid using mutagens. Molnupiravir is a drug commonly prescribed right now, but it's a mutagen and it causes a higher rate of mutation. You can also monitor and treat immunosuppressed patients who are, as we'll see later, a major source of new mutations in this pandemic. So those are tools you could use to reduce the mutation rate. You can also, on the other hand, reduce the case numbers. That's the total number of new mutations is the mutation rate times the number of cases. And reducing the case numbers is what we've focused on through this entire pandemic, through NPI measures, vaccination, contact tracing, and isolation, and what have you. So all of those tools would help to slow the appearance of an immune escape mutation. Once it appears, what do? Well, you can still, policymakers can still do something by reducing the selective advantage of the immune escape variants. One way to do that is, so to do that, you either fight the fire. The fire are those immune escape variants. So you either detect them and isolate them through contact tracing and other means, or you reduce contact rates. That's the fuel with other individuals that are prone by being previously immunized. And so we talked about the way that you can reduce that contact rate is really to try and immunize whole communities at once. So you had a few cases circulating alongside resistant and susceptible individuals for a long time. But of course, it didn't work. And almost, soon after we rolled out the vaccines, we were hit by a sweep of a major immune evasive variant, Omicron. First detected in November of 2021, and as you can see, unfortunately, that very much coincided with when the immunity had rolled out. Omicron had a series of unusual characteristics. It had 3 times more spike mutations. That's the spike mutations in spike. That's what interfaces with our ACE2 receptors and with our antibodies. So 3 times more spike mutations. It looked very different from any of the other VOCs. Many mutations were also known to reduce the ability of our antibodies to target and neutralize Omicron, and it also increased binding to ACE2, increasing transmission rate. It wasn't known at the beginning, but it also affected tropism. The mutations allowed it to enter into the cells more easily in our upper airways, and so is now largely an upper airway infection. But there were some surprises that came. So it was detected in November of 2021 that, but when we trace the evolutionary tree back through time, and this is another next strain tree with time on the x-axis, we're surprised to find that the main Omicron variants that we were sequencing diverged 6 months prior to when they were sampled, and that all of the Omicron lineages diverged from the other, here's Delta down there, from the other circulating strains almost at the very beginning, early 2020 of the pandemic. So it has this deep evolutionary history. We knew nothing about it. We didn't detect any Omicron for over a year and a half. So I want to talk and switch to how do we model this waning immunity and what does it mean for controlling case numbers now and in the future. Before I do that, I just want to give you a snapshot of what we see when we look at waning immunity at a population level. This multicolored graph is saying is from blood donors of different ages. The different colors are different ages. And when you donate blood in Canada, they look for antibodies to spike the protein in our vaccines, as well as that you develop these antibodies when you get infected. And so we're modeling this at the moment, and I can't go into details of the results, but one of the things that you can get from these distributions are the waning rates, and we saw waning over time in the end of 2021. And then you see rises in the antibody levels through boosting, both through vaccination and through infections from, in this case, the Omicron wave. That we vaccinated the older individuals in Canada right as at the start of the Omicron wave, and so this is largely immunization boost, and these later ones are largely infection boosts. So this type of model can help us project going forward how protected or susceptible we are to future waves. But this is what I want to focus on, is how we've changed, how what we see is so different, dramatically different from a year ago. When Omicron first hit, we had these enormous waves on BA1 and BA2 waves, but we don't see that anymore. Even though variants are spreading with kind of similar selection coefficients as BA2, for example, they don't, these axes are lower. And what we're seeing instead are these kind of ebbs and flows, but roughly kind of within an order of two, kind of similar levels of COVID, and just slight ups and downs, slight ebbs and flows over time. So we've seen a dramatic shift from epidemic behavior, where you can have these outbreaks and the whole population practically can get infected, mostly susceptible individuals, to this endemic behavior. And I'm using endemic cautiously because it is misinterpreted so easily. But what I mean by endemic here is that it's the amount of COVID in our community is now pretty much at a balance or steady state between an input through waning of immunity and a reduction through infections and vaccination, but mainly through infections. Importantly, endemic does not mean constant. We should see these ebbs and flows because of new variants and changes in our behavior. The other thing that endemic does not mean is rare, because the waning rate is too high and there's just too much transmission. This is too transmissible a virus to lead to the extinction of the virus. We're not going to see that. So we can, what I wanted to know was what do we expect for new variants that are rising, this XBB and 1.16.1, whatever. What should it do to the level of COVID in our communities? To answer that, I turned to a model where we allowed for waning and over time in multiple stages. And you can think of that as those antibody levels declining over time to the point where you're susceptible again. So that's what these different classes are of waning resistance. And into a model like this, we can introduce a variant, in this case, for example, one that infects earlier, that you don't have to have so much waning, but antibodies don't work as well. And so it infects, this mutant could infect earlier during this life cycle. So a mutation that did that, like BQ, spreads and has a selective advantage because it infects individuals earlier. But what does that do to that long-term rate of COVID in our community? Turns out that the answer depends on this biological feature that we don't know when a variant first appears, and that is how permanent is that immune evasiveness. If the variant infects an individual, but that elicits an antibody response that now detects that variant well, so it doesn't evade immunity in subsequent infections, that transient advantage has no effect on the long term. But there's another possibility, and that is that the variant can infect earlier the first time it infects you, and the second and the third time. And so its immune evasive properties are longer term. And we've seen that with Omicron. Omicron is not only more immune evasive the first time, you're also more likely to get it again and again and again. So it has a persistently immune evasive property. And that has something to do with immune imprinting potentially on previous variants. So we're less able to, our antibodies are less able to adapt to these new variants. The problem with that type of variant, if its immune evasive property is the same every single time it infects you, then that leads to a rise in that equilibrium or steady state level of COVID pretty much in proportion. So if it doubles the waning rate, you expect double the amount of COVID in our communities. So that's one of the main kind of features that we need to talk about and think about, and for virologists to try and estimate in order to know what the consequences will be of these variants. We're also seeing, here's some simulations to kind of drive this point home. So in both cases, so this is days, I've got different age groups, and we've got the variant and the mutation in the resident, and I've allowed the variant to have a 5% selective advantage in all of the cases that I'm going to talk about. So you see it spreading at the beginning at the same rate because of that selective advantage. But if the immune evasiveness is temporary, then it spreads, and then you've run out of susceptible individuals, and then it oscillates around, but it settles at the same place again, because now it no longer has immune evasive benefits. Whereas if it's persistently immune evasive, it retains that benefit. And you see this rising sea level of COVID cases, in this case by 66% in the long term. We have also, we can also model what would a more transmissible variant do. XBB.1.5 I mentioned was more transmissible. And so we can see it spread through a population. And in this case, we get an intermediate answer where it raises the expected number of cases in the long term, but not by so much. And in the interest of time, I think, well, this R tilde is the reproductive number, but it's the reproductive number in the population of if everybody became susceptible now with our cellular immunity and our behavioral changes. So it's it's the reproductive effect, number that we have now for the population. So the models predict, so importantly, we neither see these huge spikes in cases, nor do we see these major troughs. These are the fraction of cases just kind of bubbles up and down as new variants spread. That's what the models predict, and that is also what we see in the data now that we have a population that is that has substantial immunity. The greatest long-term risk in the future is from these persistently immune evasive variants that will raise the sea level of COVID, followed by the risk from more transmissible variants. But we're not helpless in the face of variant after variant after variant, because we know what we can do to reduce the sea level. We still have a lot of tools in our toolbox, even if we're not using them in many countries anymore. If we ramp up vaccination levels, we can short circuit this process, moving people away from that susceptibility through vaccination rather than through infection. Here in Canada, using vaccination rates we've got currently, if we double the vaccination rate, for example, we get a 40% decline in the sea level. Or we can raise NPIs, non-pharmaceutical interventions like masking and improved air ventilation. And we don't have to do that necessarily all the time. The biggest bang for the buck will be to invest in NPI measures like masks and better ventilation in those crowded areas where infection rates are highest. And if we can convince 50% of people to do that in those places where infection is a risk, and do so with effective masks or effective ventilation, this is the efficacy in a particular transmission event. Then again, we can lower the sea level of COVID substantially, depending on how effective those measures are. So going forward, I think we need to stop thinking about bending the curve, because now we are not in that zone anymore. We're in a zone where we're going to get continual input through waning of vaccines. So it's really more about what's the steady state between waning and infection. Variants will increase that steady state, but these types of measures can reduce it. And finally, in the last few minutes, I just want to talk about what do we think might be in our future? What kinds of variants might we see? And I should say that as an evolutionary biologist, understanding COVID has really shaped and changed my expectations of evolution in some interesting ways. And the first is that the variants of concern that have arisen have they've not emerged from the currently circulating strains. Alpha did not beget Delta, Delta did not beget Omicron, but in each case, their ancestry went deep into the evolutionary tree of the virus. So that was one surprise, what was going on there. A second surprise is often when those variants arose, they arose with many more mutations than you'd expect. You can see that in this graph of the number of mutations compared to the root ancestor over time. And mutations lead to a constant slope on this graph. But what happens with the new variants is often the intercept jumps up. So those variants of concern don't have a higher mutation rate, they don't have a higher slope, but when they first appear, it's as if there was a major jump in the number of mutations that they carry. So they have more mutations, like a burst of mutations happened at some time in their history. And the other thing that is of interest in these variants of concern is that they have a disproportionate number of changes in the spike protein. So they're not just any type of variant. The variants of concern, the major ones, have had all of these unusual characteristics. Here is a graph showing what mutations accumulate across the whole sequence of the genome in typical sequences. Here is what we see in variants of concern with a spike in spike. And that is more typical of persistently infected individuals or chronic infections, where again, we see a large number of spike mutational changes in that spike protein. So thinking about where's the next variant going to come from, it's worth thinking about these unusual features and understanding where they likely come from. So it's not known entirely because it's a black box where exactly Omicron was circulating, where exactly Alpha was circulating before it jumped out. But passage through immunocompromised individuals with persistent infections matches these signals. As I mentioned, it matches the signal of excess number of mutations in spike. And that is potentially because of the relaxed or altered selective environment inside that individual with a persistent infection. If they're immunocompromised, their array of antibodies, if they have antibodies, they're selecting in a different way on this virus. And that allows changes in spike that would potentially not otherwise be allowed. It also explains why these variants of concern were hidden from our surveillance efforts for over a year. And that's because a lot of their evolution was going on in individuals. And the other thing what this tells us is that there's not one evolutionary process going on for SARS-CoV-2. It's got the kind of standard evolutionary process of infection after infection after infection, but simultaneously with that kind of trunk evolutionary process are potentially hundreds of thousands of individuals that have persistent infections over time, where there's evolution occurring within those individuals, sometimes feeding back into the lineages circulating that we see. And why do I say it's hundreds of thousands, potentially millions? Well, there's estimates that 2 to 3% of the US population is immunosuppressed. And so if that large number of people worldwide are unable to kick infections, sometimes they don't even know that they're infected because they're not having the kind of immune response to SARS-CoV-2. That leads to potentially lots and lots of independent evolutionary trajectories happening simultaneously. I'm going to skip this, but this is a really great paper by Saleh et al. Showing just how within a single patient, they basically saw the evolution of a variant of concern over the time course of that patient's infection with higher immune evasion properties. The second potential source of future variants is animals. This graph just shows how many different species of animals have been infected from humans. And some of these have then come around. There's been examples of mink to human and deer to human transmission. Several sites in the genome are known to be under different selection in animals and in humans. So this again allows the virus to explore kind of a different fitness landscape, different selective landscape in which mutations can accumulate and potentially come back into humans. So I think I will skip ahead. I was going to talk about molnupiravir, but when I will skip ahead and just talk about measures that we can take as a society to reduce risk. First of all, we've got to refocus our vaccination efforts. Only 64% of the population are fully vaccinated, and the lower income countries basically have not had access yet to vaccines. We need to stop treating these diseases as separate. HIV, cancer, and other diseases that often lead to long immunosuppression are also allowing people to be persistently infected. These diseases aren't separate. And if we don't have a global health approach to all of the pandemics facing humanity, then it's not a surprise that we're going to be we're going to face again and again new variants. So we have to prioritize identification and treatment of immunocompromised people throughout the world. We have to improve surveillance. It's a good idea to avoid using a mutagen like molnupiravir. Happy to talk about that in questions if you want. As well as thinking kind of outside of the box about vaccines, getting vaccines that maybe nasal vaccines. There's a lot of work in that area so that we can stop the virus before it even gets into our nasal passages, and engage in broad risk reductions. I don't know about where you live, but there's basically a move away from any mandated or public health measure at the moment where I live. And that all or nothing, circuit breaker or nothing is not a sensible approach. We can do more as a society to lower the COVID impact on our lives and get off this roller coaster. But it requires really a much more global approach. And so I think with that, I will say thank you to all and take questions.
Wow. That was an amazing talk, Sally. Thank you so much. It was, we lived this all together so intently, right? And so to see you take all these data and turn it into the arc of what we lived through, but also then to the projection phase and the variants of concern and the public health is amazing. So thank you for that great talk. In my very brief surprise introduction of Sally, I mentioned her service to SSE. And so I wanted to thank you for your service, but I also went back and looked at our announcement, and Sally has done service for a number of societies. So I just wanted to, before we open it up for questions, let everybody know that at the meeting in Albuquerque, Sally will be receiving the official award, and we will then mention all those services. But thanks right now, Sally, for everything you have done for our field of evolutionary biology, and also for SSE. Now I'll turn it over to Mike for questions.
All right, so our first person who has a question here is Ben. So I'm gonna let you unmute yourself.
Hi, lovely talk, Sally. Thank you for that. I was wondering if new variants of concern are perhaps expected to come from immunosuppressed or immunocompromised people that they're living in for a long time. Does that have implications for what we expect those variants to be like? In particular, does that mean that there will be strong selection against killing your host, because we have all of these independent evolutionary trajectories, and the ones that kill their hosts don't end up being variants of concern?
Yeah, absolutely. So let's unpack that a little bit. Most variants that will arise are going to arise from the currently circulating strains, and what we're seeing now is tons of mutations, lots of even convergent mutational changes, and that I think is mainly these normal transmission events. There's just so much virus out there, but these kind of bursts of mutations, but also kind of variants that potentially are crossing adaptive valleys and have new combinations of mutations. Those are the ones that are that we see evolving inside persistently infected individuals. And those are what we're kind of keeping an eye out globally. Do we see signs of these kind of bursts of mutations? And you're apt, Ben, you're very astute. When I said that there was no selection on mortality, that was through normal infections, but there is selection in these long-term and persistent infections to not kill the host. Now, what I don't know is a lot of times, like it's the cytokine storms, it is the immune system that kills the host. So maybe that's not really strong selection in immunocompromised individuals. But you're absolutely right. In those long-term infections, there is selection on the virus to not kill. And even to not be so severe that you go to the doctor to clear it.
All right, next up, we have a question from Sarah.
Hi, that was awesome. I read so many papers, but to see it in 1 hour put together was really helpful. I have one biology question and then one question for you as someone in our community who's interfacing with policymakers, because we just don't get to hear from people doing that much. The biology question is, those immune evasive variants that you talked about, are those mostly immune evasive because of properties of the viral particles? Or is it something related to it sounded like you were describing something like antibody exhaustion or antibody change with repeated infections, or is it just both? I just hadn't heard about that change in antibodies and how they react to multiple infections. So if you could explain that to me, or maybe I need to take an immunology course. And then I have another question about policy.
I think that's a fantastic question, but we seem to have lost Sally from the Zoom call. So
maybe she'll come back.
she'll be able to get back in a moment, but at the meantime, I'm going to unmute Michael because I think Sally has the option to go through him instead.
I don't know what just happened, but fortunately I have a second computer in the house.
Do you want me to repeat the question,
Yes,
Sally?
please. I didn't hear his name.
So those immune evasive variants that you talked about in the first half, I'm really interested in that. And I've always thought of that as being a property of the viral particle and the mutations it's accumulating. But you said something that made it sound like there was something that happens to antibodies and their ability to, and that's independent of the virus and the variant.
Yeah.
Can you
it's definitely a dance between our bodies and the virus. So the B cells that produce the antibodies, when we first get exposed to the virus, that B cell repertoire that recognizes the virus expands. And it expands recognizing the particular virus that it's first exposed to. And it's called immune imprinting. And so subsequent infections with a slightly varied virus trigger an immune response, but that's mainly the response of the B cells that we already have. And so the B cell repertoire can fight the new infection, but without actually being very good at expanding the B cell repertoire, because it's able to fight it and clear it without detecting the change.
So that's kind of totally, go ahead.
So that's sometimes called oh dang it, an immune antigenic sin, but that's kind of seeing it in a bad light. Really, it's an amazing immune system that is able to respond really quickly to something similar to what our bodies have seen in the past. So immune evasion is partly that it's different enough from that B cell repertoire in each individual.
It's kind of counter to the conventional thought that if you get infected over and over again, you get the benefits of building immunity, though, that this almost sounds like your immune system gets depleted by these multiple infections.
No, I don't think, I think this expansion of your B cell repertoire happens with every infection.
But it's not specific anymore.
Well, it is, yes, it gets increasingly specific. So, and even these bivalents, it helps a little bit by kind of teaching a dog new tricks. Yes, you work it hard enough and it will learn new tricks. But I should say that every single infection comes with the risks, the risks of long COVID symptoms, the risks of infecting somebody that's particularly at risk of severe disease. So there's some individuals that say, oh yeah, you want to get infected to train your immune system, and I'm not one of those proponents.
Yeah, that was I feel like a huge part of the US messaging, the anti-vaccine and anti-masking messaging
Right.
was like, like, let's build this herd immunity, and that suggests that that's not
No.
going to work. Can you talk to us a little bit more about your experience working with policymakers and the reception you got and their appetite for understanding what's going on under the hood using these models versus, I mean, just we never get to hear about this.
Yeah, it mixed bag, mixed blessing, and I think probably my colleagues in the U.S. have similar experience, because sometimes there was I would be working with people that didn't really want to know what the models would have to say unless it agreed with what the policy that they wanted to change. So there were some that was very, very hard when it was, the models are not consistent with what you're saying. This is not evidence-based, what you're saying. And so I would speak out, and then that would make them in the press, and that would make them angry because I hadn't kind of towed the line. And so there was a lot of tension, especially locally. At the federal level, it has been really positive. There's been an interest in knowing, getting guidance about what might be coming down the road, what we could possibly do to lower infection risks. Canada took advice from me and a few other scholars on molnupiravir. Molnupiravir is this, it's a base pair analog, and during the replication of the RNA virus, it goes in, but then it can't be replicated properly, and so leads to a mutation. The idea was this error catastrophe idea from evolutionary biologists that if we just riddled viruses with so many mutations, they'd have no offspring. Sounds like a great idea. But when you looked at the data, the virus was still replicating, and it wasn't riddled with mutations. It just had a few. And so it was like this scary zone of, we're basically adding mutations to this virus and potentially allowing it to evolve faster. And we're starting to see emerge signals of more rapid evolution in those countries that use molnupiravir. Fortunately, Canada did not. And I think, France did not approve it either. So there are some countries that I think took the evidence and the concerns about the pandemic when deciding about the drug. And that's actually, I think, a super interesting question. When drugs get approved or not approved, it's about the individual patient. That's what governs the decision about whether or not a drug should be approved. But in a pandemic context, you should also think about what are the consequences to others.
All right, we got a question that scrolled through the chat from Kurt. And so Kurt, I'm giving you the option to unmute yourself and ask your question here.
Shall we read it, or are we?
I think I can read it. And I think the question was, is it possible we've got the root wrong? And normally when we do phylogenetic trees, I'd say yes, there's a real possibility of that. But we've got so many samples at the time that there's not that much uncertainty in the position of the root. So no, I don't think it's possible that the Omicron variant is at the root of the tree.
All right, we've got a question here from Leanne. So Leanne, go ahead.
So I'm wondering, with the ubiquity of home tests now, if you're seeing more minor variants that maybe aren't as intense in terms of their symptoms, showing these longer root branches or these longer branches where you're having a lot of evolution before you're actually sequencing this variant because it's circulating in the population and only being tested at home.
Absolutely. And we've seen just this dramatic decline in cases among the under 70-year-olds in Canada. So basically they're not being tested. It's almost impossible to get a test. And so the virus has to jump from patient to patient to patient until it is in one of the classes of people that that are eligible for testing, which in Canada at the moment is PCR, and a lot of those are sequenced. But I think I don't know if you were asking, but there's some other, some people have speculated that there may be selection to avoid, to change where the rapid antigen tests are detecting, so that people aren't aware that they're infected and continue to circulate out. I haven't seen any evidence that selection to avoid a positive rat has occurred so far, but it's possible.
All right, we had a hand up briefly from Joanna. If you want to ask a question, feel free.
Sally just answered it before I asked it.
All right, that's a thing that happens.
I have a question. This is sort of more of a personal question, Sally, but what are you going to do, now that we sort of know that we're in this like seascape of
flows, yeah.
What are you going to do next?
Well, it's really this CoVarNet to have a group of people that are working together and to automate it so that some of this kind of automatically we can so but I think we need to look forward to getting improved processes, and we've had so much with data in this country. We protect the privacy of this bloody virus, and it drives me crazy. So there was never anybody in Canada, in the entire country, that had the genomic sequences alongside with whether those individuals died or lived, whether they were vaccinated or not. So we had all of these kind of independent data sets, but we weren't trying to put it together to learn. And so that's one bee in my bonnet, is to really work data information systems and connections so that we're better prepared to learn in the future, that we can link the kind of data sources. And that's true for COVID, but it's also true for species at risk, where we have all sorts of these different piles of data, and we're spending lots of money and not pulling the data together. So that's one thing.